Hello everyone. Let's talk about the immune systems of fish. Why do we care? One reason is the aquaculture industry now accounts for about half of the world's supply of fish. But concentrating fish in pens like these make them highly susceptible to infection. So we have a need to understand their immune systems better, and we also think we may learn more about our own immune systems from studying them. When one of us is infected with a pathogen like COVID-19, our body defends itself by producing an immune response, including the production of antibodies. Much of what we have learned about the human immune response comes from studying the mouse. Humans and mice share over 60% of their genome, and their immune systems function very similarly. Both of us produce our antibodies inside our spleens within immune cell clusters we call germinal centers. We can stain these germinal centers with specific reagents which help us visualize what's going on inside. Germinal centers increase in size when infected, so we use size as a marker for immune system activation. But what about fish? We can breed these stickleback fish easily in our laboratory and work with them to locate their immune centers and learn more about how they function. We suspect that fish immune centers are also located in spleens, and though we don't have species-specific reagents to stain them, they are naturally pigmented, and that's why we call them melanomacrophage centers. In order to determine whether these suspected immune cell clusters grow larger in response to infection like germinal centers, we're going to do an experiment. We're going to divide our fish into two separate treatment groups. One group we're going to inject with a protein that simulates an infection. The other group we'll inject with a controlled substance that shouldn't have much effect at all. And every 7 days, we harvest spleens from several of each group. We freeze the spleens and put them through a slicer and look at the thin sections under a microscope. We use image analysis software to measure and compare the size of fish immune cell clusters for each treatment group. Then we plot the results. Here you can see that from day 7 to day 14, the control group doesn't change that much in size. But after 14 days, the immune clusters of the treatment group have grown significantly. Now we know that just like mice and humans, these fish immune clusters grow larger when treated with a protein. And now we know that there is an evolutionary continuum for the location and function of vertebrate immune systems, and that gives us a base point for further studies, which is good news for our stickleback fish and the future of aquaculture.